Hello students, hope you are safe and well. We look forward to having you back on campus. Now, here we look at the third week of development and some interesting events happening here. This is also known as the week of threes, where you have important processes like gastrulation, notochord formation, and neurulation occurring, as well as the early vascular system is established. Now, last time we saw how implantation was complete, so now we are moving the story on from there. So these are the objectives for the lecture. We are going to see what is this primitive streak and its function, how it's essential for the process of gastrulation and how it establishes the body axis. And in gastrulation, we will see how the bilaminar germ disc becomes a trilaminar germ disc and the three embryonic germ layers, that is the ectoderm, mesoderm, and endoderm, are formed, and what is their fate in the next the fate will be seen in the next class. What is notochord and why is it necessary? So its significance and what happens to it later on. Then how is the early brain formed or the neural tube formed by the process of neurulation, and how an early vascular system is established. Okay, so all this is happening in this important third week of intrauterine life. So this is review from the last time. Remember the bilaminar germ disc had developed, and that was the week of twos in the second week of intrauterine life. So this picture shows in a summary that's blastocyst implanting, and then remember the differentiation of the trophoblast into the syncytio and the cytotrophoblast, inner cell mass, the cavity developing inside, that's the amniotic cavity, the bilaminar germ disc made up of epiblast and hypoblast, the blue and the yellow cells, the original blastocyst cavity becoming the first, the primary yolk sac, then the secondary yolk sac by cells multiplying from the hypoblast and surrounding the original yolk sac, and then how the extraembryonic mesoderm developed and the splitting of it and the development of the chorionic cavity and the chorion as seen in the last picture below, right? So let's move on. So this is just to orient you as to where all this was happening. So remember that morula had come into the uterine cavity, become the blastocyst, which came in contact with the endometrial wall. And you remember the normal sites of implantation. Now this is a schematic picture. So this black dot is telling you what is happening inside that blastocyst, etc., that we just saw previously. And see now at the end of the second week of development, it has got buried completely inside the endometrial layer. That is that red layer seen in the black dot buried inside. The endometrium has grown over it. So there's nothing in the uterine cavity, as if there was nothing. Now, if you look at the second picture, it is an enlarged picture showing you inside what's happening, which we've already seen. And you can see the original endometrium has grown back over the implanted structure. So there's a slight raised area where it was implanted. Now, just to realize or understand how the endometrium is named according to where this implanted site is. So, towards the inner cell mass area where the original blastocyst implanted, remember we said the endometrium there undergoes the decidual reaction, and where the syncytial trophoblasts are invading, etc. So that part of endometrium is known as the decidua basalis. Now this will form the future maternal side of placenta. We'll understand this later, but basically it's like a nourishing bed, right? Now, the part of endometrium, that epithelium which recovers that implanted blastocyst, is now known as decidua capsularis. Okay, and then come back to the first picture. So, decidua capsularis will be just above that, covering the black dot, and the rest of the endometrial lining, which is lining the whole uterine cavity, right, is now known as decidua parietalis. So, do you understand? At the site of implantation, the endometrium is known as decidua basalis. Covering the implanted structure is capsularis, and the rest is known as decidua parietalis, or lining the wall of the uterine cavity. So this tells you where we are, like a Google map. Now let's see this new event happening in the bilaminar germ disc. Remember, we are in the third week, so at the beginning of third week of development, we can here in this figure see this germ disk. Now we are seeing the top layer. That means it is the epiblast layer, right? The hypoblast would be below that if you look at this germ disk from the top. Now here you're not seeing all the other things happening around. So we are just focusing on the germ disk as if we are seeing it from the top. So at the caudal, at one end of, now that we don't know caudal and cranial end initially, but at one end, which is going to be the caudal end of the germ disk, some cells start coming together. So they multiply and come together, form a longitudinal column of cells known as the primitive streak, which starts growing towards the other end, which becomes the future cranial end. So you understand that this primitive streak will develop in the midline in the epiblast by heaping up and multiplying cells, always from the caudal end, the future caudal end of the embryo, right? And it starts growing towards, so the other end automatically becomes the cranial end, right? Now, as this primitive streak grows, at its cranial end, it grows to a certain length. At its cranial end, again cells heap up to form a rounded structure known as the primitive node. Now, in that node, imagine you are creating a pit, a depression. So that is known as primitive pit. And in this original primitive streak, also you are creating a depression um, called the primitive groove. It's like playing in the sand. First, you make a ridge, like a raised area, then you make a rounded area at the end, then you make pits and grooves inside that, right? So, from where have these cells come? Cells in the epiblast have started multiplying. Okay. Now, what these cells will do, these are very hyperactive cells, they will start moving, migrating somewhere else. So, let's see where they will go. So again in the upper picture, you can see the germ disk, and the arrows in the blue picture above indicate that, okay, the midline structure is the primitive streak, and then the arrows indicate that cells from this primitive streak will start going inside the primitive pit. See, they are actually going through the primitive pit and groove, and which cells? Now cells from the epiblast. They keep multiplying, and they will enter this primitive pit and groove and come down below the epiblast. So in the middle second picture, the blue layer is the epithelium of the epiblast, right? So we were looking at the germ disk from the top in the first picture. Now through this hole and this groove, you can see the red cells migrating below the epiblast, right? That means they migrate ventrally or inferiorly by invagination through primitive pit. And of course, these are controlled by all genetic factors. So FGF8 is known as fibroblast growth factor, and this is produced by primitive streak themselves. So the primitive streak has, in short, appeared at the caudal end of the germ disk in the epiblast. It has grown cranially, and now cells from the surrounding epiblast start moving towards the primitive streak and pit and start invaginating downwards or ventrally through this groove and pit. Is this fine? And the third picture is actual picture of the embryo showing you that primitive streak and heaping up of cells and then the pit and the groove in the midline. Okay. So that means why is this happening? Now the primitive streak then is essential for the process of gastrulation. And this is defined as the process of formation of a trilaminar germ disc or the three germ layers from a bilaminar germ disc. So you can see some cells in the second picture are invaginating in between like a sandwich, and then hence in future you will get three layers. We will learn to name them. So the what happens to the first few population of cells? You can see in the second picture the red cells, they're going straight down, and remember the yellow cells were the hypoblast, right, in the bilaminar germ disc. So the first few cells migrate into the hypoblast and start replacing that original primitive hypoblast cells, and henceforth they will be known as the embryonic endoderm layer. So that means the endoderm cells, I mean the cells from primitive streak, come into the hypoblast, replace the original hypoblast, and then will be known as endoderm. Okay. Other cells move laterally as shown in the image again and will form a middle germ layer that becomes the future mesoderm or intraembryonic mesoderm. Why intraembryonic? Because we are in the germ disk, right? And the cells that will remain, original cells that will remain on the epiblast now, including the blue cells like that, will now be known as ectoderm. So from a two-layer germ disk, we have three layers made up of endoderm, mesoderm, and ectoderm. Let's look at more pictures. So in this again, you can see the same thing happening. Now the orange, if those of you who can't see the pointer, so I'm describing the colors. The orange layer, upper layer is the epiblast. The green cells are the migrating cells from epiblast. You can see first they are trying to replace the yellow cells of the hypoblast. They will slowly replace everything, and then this will be the endoderm. And then the green cells migrate laterally also and will become the mesoderm. And whatever is above is remains as the ectoderm. Okay. Again another picture. So you can see again now, look at the red cells. They are spreading laterally, right? And these are forming the mesoderm. It's also known as intraembryonic mesoderm because this is happening within the bilaminar germ disk. Okay, that means cells migrate downwards. That means ventrally, laterally, and cranially. You can understand in the second picture they will go cranially like that and totally become the second layer, middle layer between the original hypoblast and the original epiblast, as shown in the lower picture also. Okay, what happens at the margins of the embryonic disc? That means once these cells have filled up everything in between the epiblast and hypoblast, at the margins you can see these cells will merge or will be continuous with the extraembryonic mesoderm that is already there surrounding. And to give you the orientation, the cavity above the blue cells was the original, the amniotic cavity developing, and below was the yolk sac, and surrounding is the extraembryonic mesoderm. So the cells just become continuous there. Okay. Again, this picture, you can see the arrows. That's what we meant. Migrate ventrally, laterally, and cranially. And laterally, they become continuous with them. So here we have the three germ layers finally formed: the ectoderm, mesoderm, and endoderm. Why this is important is because through these three germ layers, all our tissues and organs and systems will arise. So this whole process of formation of three germ layers is known as gastrulation. Okay. Now, this primitive streak is important for many reasons, as it is giving rise to the mesoderm cells which are migrating in between the epiblast and hypoblast. Some of the early cells from this primitive node migrate in the midline and cranially form the precaudal plate. It's just a collection of cells. Now, cranial to the precaudal plate is an area known as the oropharyngeal membrane. So it's a great organizer of different regions too. Now at the oropharyngeal membrane, there are no mesoderm cells migrating in between the surface epiblast and hypoblast. That means in this region, the ectoderm will be in direct contact to the endoderm. Now this oropharyngeal membrane represents the future external opening of the oral cavity. That means this membrane will break down in future, and our oral cavity then will be connected to the outside. Similarly, caudal to the primitive streak, an area forms that is known as the cloacal membrane, where again there is no mesoderm migrating in between the ectoderm and the endoderm, which are in direct contact. So that's the area of the future anal membrane or cloacal membrane, which will break down. Hence, our future GI tract will be continuous with the outside environment at this area. So there is no mesoderm where at the oropharyngeal as well as at the cloacal membrane site. And the precaudal plate is just a orientation area because the oropharyngeal membrane will be located just cranial to it. So in this upper picture also you can see both the membranes. Okay. So this picture, don't worry about these short forms in this image. This picture is just telling you that cells from the primitive streak are migrating in a very organized manner, not randomly, giving rise to intraembryonic mesoderm, which will also have specific parts. So just to know, PM stands for paraxial mesoderm, intermediate mesoderm, lateral plate mesoderm, etc. This you will learn later, but it is mesoderm basically, and is migrating in a very organized manner. So they migrate everywhere, as we said, ventrally, laterally, and cranially, except where? Except in the region of the oropharyngeal membrane, in the region of the cloacal membrane, and where this third structure, can you see that structure, blue structure named N in the picture in the midline? A head or cranial to the primitive streak, that is the notochord. So there also there will be no mesoderm because notochord, this structure, cord-like structure has to develop in the midline. Why, we will see, right? So these three places, as noted, there is no mesoderm in between. So after we have looked at the process of gastrulation, now we see the formation of another structure known as the notochord, and we will see its importance, right? Now remember, all this is happening simultaneously. So in this lower picture, you can see the primitive streak with the groove, the primitive node with the pit, and the three germ layers have formed. You can see the three colors in the lower picture, the upper ectoderm, mesoderm, and the endoderm, right? And below is the yolk sac, and above this will be the amniotic cavity, which is not shown in the picture as if it's cut. So this is the three-layered trilaminar germ disc, as it's called. Now, while the primitive streak is giving rise to, these cells are migrating through the primitive streak to give rise to the mesoderm. And note another thing happening. A collection of cells from, again, the epiblast only. Remember, the blue is all epiblast. Some cells don't go to form the mesoderm, but they will go down below the epiblast from the primitive pit. They invaginate downwards or ventrally, and now but remain in the midline and grow cranial to the primitive pit. That is shown by the dotted line extending towards what? Extending towards the oropharyngeal membrane, but they don't go beyond the oropharyngeal membrane as seen in the picture, right? Now, what is this group of cells known as? Initially, as the cells get together, it's called a pre-notochordal plate, and then it soon organizes itself into a notochordal process formed in the midline. See, it is shown in dotted line because it's beneath the ectoderm or beneath the epiblast cells, right? It's not on the same plane. The cells have migrated beneath but remained in the midline. In the upper picture also, the same thing is shown. You see that arrow going up in the midline towards the oropharyngeal membrane or the buccopharyngeal membrane. It's also known as buccopharyngeal membrane. So that is the prenotochordal plate which will form the or prenotochordal cells, and this will become the notochordal process. Now let's see what happens to this process. So this is first picture is just telling you the cut section of the second, third, and fourth picture. So it's in the midline as if you're cutting the embryo sagittally, into a half. So in the first picture, you can see the primitive streak, and the dotted line cranially represents that notochordal process. Okay. So in the second and all the other pictures, the color red is showing you the solid notochordal process which has grown downwards. From the primitive pit, cells have grown down below the ectoderm, right? In the midline. So what is happening soon to this solid plate? It's a notochordal process, solid plate of cells. You can see it soon starts getting canalized. See that notochordal canal developing in that solid plate, and look at the third picture, the notochordal canal is named, right? So that is the next step to happen. So first was a prenotochordal plate which became a notochordal process cranially, it grew cranial to the primitive streak towards the oropharyngeal membrane. So in section you can see it as the red color, and then this started to become canalized, means some cells are you know undergoing death there, and it becomes a hollow structure which is known as the notochordal canal. Okay. Then next what happens? So this is best learned by pictures. Now this notochordal canal, remember it's a hollow tube, so it has a roof and a floor. So imagine now the floor of the canal, which should have been red here, okay, but this floor of the canal fuses with the underlying endoderm. See, yellow is the endoderm, right? And then it breaks down. The floor breaks down. So what do you have? You have a notochordal plate present. Okay, means now only as if only part of the roof is present. So again, it becomes a plate. And because of this breaking down of the floor, you can see in the next second picture, a temporary connection is formed between the cavity above, which is the amniotic cavity, and the yolk sac. And this connection which is passing through the primitive pit is known as the neurenteric canal. Why? Don't worry about it now. Neurenteric means digestive system, because this part of the yolk sac is going to get absorbed in the embryo to form the digestive system tube. It's called the neurenteric canal. It's a temporary opening. Afterwards it closes. Okay, now what happens to this plate? Remember whose flora had broken down. So in section, in the lower two pictures in the section, you can see that is the plate, notochordal plate labeled there, where its floor, remember it was a hollow canal. Then you can imagine in section the floor breaks down and then it becomes a plate, only the roof remains. And this plate then again, now see, becomes a solid cord. Now it has no lumen, no canal, no cavity, and then it's called the notochord. So you saw the processes that happened. First was a pre-notochordal process, then notochordal process, then notochordal canal, then the flow broke down, notochordal plate again, and then that plate again becomes a solid rounded notochord, right? And then that which regrows like this again cranially completely, it becomes a solid notochord, and that neurenteric canal closes again. Okay. So here again in the same picture, you can see the red, that was the notochordal process, became the canal, the floor fused with the endoderm, so it became a plate again, and then imagine it's becoming a solid cord. Okay. So where does the notochord extend from? It extends from the primitive pit up to the oropharyngeal membrane, but not beyond it. So see in section you can make out how the oropharyngeal membrane and the cloacal membrane look at the cranial and caudal end of the embryo. You can see there is no mesoderm in between. So just ectoderm and endoderm stuck together, right? Now why is the notochord necessary? This solid cord, remember it's in the midline again, is going to form the basis of the axial skeleton, our vertebrae and vertebral column and our axial bones, right? So what is the function of the notochord? It has many functions. Firstly, it develops in the midline. It defines the primordial axis of the embryo, just like the primitive streak is in the midline. But now it's a solid cord. It provides rigidity to the embryo. It serves as a basis for the development of the axial skeleton. The vertebrae will grow around this. So first the notochord must develop. Imagine this cord like that, and then the vertebral bodies will surround it and will grow. Okay. So it serves as a basis for the development of the axial skeleton. It stimulates the formation of the neural plate in the ectoderm. So only once that's the next step that we're going to see how the neurulation happens. So only if the notochord develops normally will the cells above in the ectoderm then further differentiate to form something known as the neural plate. That is the early beginning of development of the nervous system. Okay, so it induces the formation of the nervous system or the neural plate where in the ectoderm above it, not in the mesoderm, in the ectoderm above it. Then it also indicates the future site of the vertebral bodies in the column, because I said the vertebral bodies will grow around it, will ossify around it. And what else does it do? So it kind of gives an access to the embryo, right? So it also regulates the differentiation of other surrounding tissues, including, as I said, it stimulates the overlying ectoderm to form the neural plate. It helps to differentiate, helps the mesoderm to differentiate further. Okay, so these are the many functions of the notochord, mainly providing axial rigidity and providing the basis for the axial skeleton to develop around it. Note at this time, we are not going to understand why it's happening, but because it's in the picture, note this. This is the secondary yolk sac, right? And note one little kind of a pocket-like structure, a diverticulum or a growth growing into the future, that connecting stalk area. And this is known as the allantoa or allantoic diverticulum. Why and all that significance, don't worry about it now. But this is what is also seen in the picture. Okay. So now you can understand amniotic cavity, yolk sac. Now here remember the outer, that chorionic cavity and all is not shown here, but it will be there, right? In the second week that had developed. So we are only concentrating on what's happening in the germ disc right now. So what happens to this notochord once it's formed? As I said, around that the vertebral bodies will start forming. So it induces cells around. Basically, they are the mesodermal cells around to form the vertebral bodies and the vertebrae, etc. And once all this vertebral column is formed, so vertebral column is separated. I mean, the bodies are separated by the intervertebral disc. Do you remember the structure of an intervertebral disc? So outside is a thick fibrous ring, right? Strong ring that is known as annulus fibrosus, made up of fibrocartilage, able to withstand compression. And inside is like a soft uh, jelly-like mass almost, that's known as the nucleus pulposus. So this inner mass, the soft mass, is the remnant of the notochord. So what happens is once the vertebral column is formed, the most of the notochord will degenerate and disappear as the bodies develop, but only part of it that lies between the vertebral bodies persists as part of this intervertebral disc. So the nucleus pulposus actually of each intervertebral disc represents the original remnant of the notochord. Otherwise, it's supposed to disappear. But like any tissue, like any embryonic tissue, if it persists, it can, form kind of embryonic tumors, and these are known as chordomas. And the most common site is usually at the base of the skull, here or here in the second picture that is shown here. And in the third picture, the picture marked A and B is showing you a big mass attached to the spine, but it's lower down. So even sometimes you get it in the sacral, lumbosacral region, and it will be made up full of kind of early embryonic tissue or notochordal-like tissue. So then you can understand it may be benign or may turn malignant later on. Anyway, it has to be removed if detected. So this is where we were again, summary, the same picture. So we started with the primitive streak, remember, which generated the intraembryonic mesoderm between the ectoderm and endoderm, the three germ layers. So gastrulation happened. Primitive streak first happened, then gastrulation happened. Primitive streak determined the craniocaudal axis because it developed at the caudal end. Then we saw how the cloacal membrane and buccopharyngeal or oropharyngeal membrane developed, and then we saw how the notochord developed, right? So what is the entire function of this primitive streak and what happens to it? We'll see this before we see what is this neural plate and all. So what are the functions of the primitive streak now? As I said, it determines the craniocaudal axis. It determines the bilateral symmetry of the embryo because it appears in the midline, and it also determines the dorsoventral surface of embryo because now ectoderm is dorsal and the cells migrate ventrally. So whatever is down, right, is ventral to the epiblast. So dorsal ventral orientation is determined in the embryo, and it gives rise to, it is the source of the intraembryonic mesoderm, so it helps in important for gastrulation. It induces the formation of the notochord beneath the ectoderm, and lastly we are going to study this now, how it induces the formation of the neural plate in the ectoderm, which is here, that plate that is labeled in the figure. So is that clear so far? Now, before we see neural plate formation, let's see what is the fate of this primitive streak once it has performed all these functions. So the primitive streak, now you know, is essential for gastrulation. It's essential to induce notochordal formation, neural plate formation, etc., etc. Now, once it's done its job, that means even the neural plate and neural tube will be formed, brain is starting to form, and notochord is completely formed. And the third picture you can see. Now see the downward arrows of the primitive streak, the pink color, and the white arrows. This is indicating that it starts regressing. So after it finishes all its functions, it starts regressing and degenerates after fourth week of intrauterine life, after gastrulation and neural tube formation and uh, notochord formation is complete. So just about in a week, see, it has done its job and it starts to degenerate. It will become smaller and smaller and smaller and it will disappear finally. But again, like embryonic tissues, if it fails to degenerate completely, if part of it persists, again, it can give rise to, because remember the cells are very, very pluripotent, primitive streak cells are, that means like stem cells. So they can continue dividing and give rise to all sorts of germ cell tumors, which is known as a teratoma. And if you take biopsy of this, it will have derivatives of all three germ layers. You might get Any kind. You might get gland tissue, connective tissue, teeth, bone, any mix up tissue. That's why it's like a teratoma means monster. It might be like any mixed up way, weird kind of tissue. Teeth may also form inside this mass. So it most commonly you can see it in the sacrococcygeal region. See how huge in this upper baby or even in the lower baby. And it is called a sacrococcygeal teratoma, which can result from the remaining primitive streak. See, it regresses from cranial to caudal, right? If cord leaf part of it remains, which can become a big tumor like that. They're usually benign, but see how they can grow. And once you remove it, the prognosis is good. They can be removed. Only thing is, if they grow so big, you see, they might displace the anal bladder opening or anal opening, and that will have to be corrected because of its location in the lower region. It might obstruct. All that will have to be corrected. So for some reason, it's more common in female babies, and of course, the treatment is surgical removal. Okay, so this is what happens if embryonic tissue, which is supposed to regress or persist for some reason. Obviously, it's a genetic mutation. So lastly, in this important third week of intrauterine life, let us see what we mean by neurulation. Simply, it means formation of the neural tube. Now, where is this all happening? So see, again you recall primitive streak is doing its function, giving rise to mesoderm. It has given rise to the notochord. Now, under the inducing or stimulating effect of the notochord, remember this notochordal process is lying beneath the ectoderm. The overlying ectodermal cells, overlying, okay, thicken again, form a thick plate known as the neural plate. It's like a slipper shape. Look at the shape. It's like the sole of your shoe or a slipper shaped neural plate. Okay. Now where does that develop? So again it develops cranial to primitive node. Again it extends up to oropharyngeal membrane, not beyond that. Okay. So let's see what happens to this neural plate. This is all happening simultaneously in the third week of intrauterine life. So let's see how this neural tube is formed. Now, these are sections of the embryo at, so let's look at the sections, right? Now, remember the blue is the ectoderm. So first the neural plate is formed in the ectoderm where just above the notochord. So you can see in the first section, notochord is labeled as a red solid dot, and that has induced the cells above to form a thickened plate, neural plate, right? Then this plate, now you can see a groove appearing, like a depression appearing in the neural plate in the second picture, and that is known as the neural groove. That groove in the third picture you can see is deepening, deep depression, and hence the sides are raised. So the sides are known as neural folds. And then what will happen in the fourth, you can understand from the picture itself, these two neural folds have fused together and a neural tube is formed. And this neural tube then will sink below the surface, and this original ectoderm will grow back as if nothing existed. So that's what you're seeing here in the last picture, that the neural tube has gone below the surface and then it is grown back again, right? The ectoderm has grown back. So in sequence, in the midline, what do you have? Do you have mesoderm in the midline? No, in this case. Here we have the ectoderm, then we have the neural tube, which is a hollow tube, and below that you have the solid notochord around which the vertebral column will form, and then you have the yolk sac, and on the sides you have the intraembryonic mesoderm. Is this okay? So this is how the neural tube basically forms from the neural plate in the ectoderm. Again, this is a picture showing you the neural plate, the neural groove and the neural folds, and then the neural tube. But note now what is happening before the two neural folds fuse. So can you see some green cells that are at the top of the neural fold are known as neural crest cells. That's why they are called crest. Crest means the top of a mountain, the peak of a mountain. So since these cells are at the top of the neural folds, they are called neural crest cells. Before the folds fuse, the neural crest cells break off. They start breaking off from that neural tube and they start migrating. They're breaking away and they migrate to different areas of the embryo. They will give rise to varied structures in the body. Okay. They migrate laterally and go to different sites in the embryonic disc. So this is the way the neural crest cells break off and neural tube forms. Okay. This is another picture of the neural tube forming in the cross sections of the embryo, and you can see the neural crest cells, and they are breaking off in the third picture and will migrate to give rise to many things. And the neural tube then, once the crest cells break off, only then the neural folds fuse, and then the neural tube forms and sinks below the surface. This fourth picture of the embryo is actual see neural tube and the solid notochord below that, and overlying surface ectoderm has grown, neural crest cells breaking off and migrating. And this is the fourth picture on the right is just showing you, the black arrows are showing you migration of neural crest cells. You can see you've done the development of the pharyngeal arches, right? See, they're migrating in the head region, face region, into the pharyngeal arches and throughout the body. So what do? The neural crest cells? Here is a list. Now you cannot make head nor tail at this stage why they form all these varied structures. But here is a list of some of the derivatives of neural crest cells. Finally, what are they going to form? They're going to give rise to some of the connective tissue, bones of the face, skull, all sensory ganglia, the dorsal root ganglia on the cranial and spinal nerves, some C cells of the thyroid. They even contribute to the spiral septum, the aortopulmonary septum in the heart. You learn this with heart development, something in the teeth, dermis in the face, neck, all autonomic ganglia, adrenal medulla, because that also has ganglion cells, Schwann cells, satellite cells, or the neuroglial cells. Leptomeninges means the pia and the arachnoid. So these neural crest cells are going to contribute to all these structures, melanocytes, and some smooth muscle cells of blood vessels, especially of the face and or brain, etc. So they are finding some tissues in the eyes also. They contribute to some of the layers of the eyeball. So there are varied kind of derivatives. The only reason we can think of is maybe all these tissues need to develop early because they have some important functions. So they are developing from the migrating neural crest cells. Okay, or they have to keep some contact with the outside world. So this in the embryo, we were looking at it in section. This is the actual little embryo where the neural folds you can see are fusing. The neural tube begins fusing to form, neural folds begin fusing to form a hollow neural tube. Now, where does the fusion begin? It begins in the future in the cervical region and then extends both cranially and caudally direction. Okay, and so obviously, if it starts somewhere in the neck region, you can imagine you yourself. If it starts in your neck region, the fusion of the folds. Remember, it's a whole line tube and groove, and the folds are longitudinal like that, so they have to keep on fusing, right? So initially then this neural tube is open both at the cranial and at the caudal end. What are those areas called? They are called cranial and caudal neuropores. Pore means opening, or anterior and posterior neuropores. Now they close in a very precise manner. The cranial neuropore closes at day 25 of development. So where are we? We are in the third week, right? And caudal closes 3 days later at day 28. So first, which one closes? The cranial neuropore closes. Okay, and this neural tube then is the precursor, or this is going to give rise to all parts of the central nervous system, the brain and the spinal cord, right? So the caudal 2/3 of the this neural tube represents the future spinal cord. The cranial 1/3 of the neural tube represents the future brain, which can be divided into forebrain, midbrain, hindbrain, etc. We learn that with brain development. So what can happen now? This tube must need to close completely. It must become a closed tube like this. What can happen if this either the cranial or the caudal neuropore opens? Doesn't fuse or remains open, then it can result in neural tube defects. So let's see examples of some neural tube defects. Okay. Now let's see some examples of neural tube defects. So any disturbance in the process of neurulation can give rise to various types of neural tube defects, which may involve either the brain or the spinal cord. Now most of these defects occur you can understand now from the process of neurulation, they will occur due to non-closure or defective closure of the neural tube. So if this defect involves the cranial region of the neural tube, that means parts of the neural tube in the upper portion fail to fuse, then you can get defects of the brain. An example is anencephaly. We'll just see it. If the defect involves the lower parts of the neural tube, then we can get defects of the spinal cord. Example, spina bifida. Okay. Now let's see here. If the cranial neuropore fails to fuse, then you will get brain defects where you might get partial or total absence of the brain itself or parts of the meninges, or usually these defects are also accompanied by skull or bone defects. So as you can see in these pictures, the part of the head or brain tissue also is missing. This is known as anencephaly. Cephaly means head, brain, and NN means absence. As you can see, these will not be compatible with survival of the fetus. So failure of fusion of the cranial neuropore can result in examples of anencephaly. Now, if in this lower picture you can see again a baby born with a kind of a soft cystic swelling. What does this mean? This is also a type of neural tube defect where there is a skull defect and meninges may herniate out. If only meninges come out, then it's known as a meningocele. But if brain tissue also comes out into that swelling through the bone defect, then it's called a meningoencephalocele. We'll see more pictures there. Now, usually these neural tube defects are associated with folic acid deficiency. Hence, women of reproductive age, you must be aware, are given usually regular doses of folic acid to prevent neural tube defects. Now, one of the signs or an antenatal diagnostic test of a strong risk of neural tube defect is there is a high level of alpha fetoprotein in the amniotic fluid. This is a substance produced by fetal cells. Again, more of this you will see when we do antenatal diagnostic tests later on. Okay, so this is just an association found with folic acid deficiency and a high level of alpha fetoprotein in the amniotic fluid. So here are some examples again. If the defect involves regions of the lower part of the neural tube, so there will be resultant spinal cord or even vertebral column defects. So usually they are accompanied by vertebral defects where the vertebral bodies or spines fail to fuse. Okay, so you can imagine this whole neural tube here must fuse, right? But if any part, lower part or anywhere fails to fuse, you will get spinal cord defects. So let's understand what we mean by spina bifida, meningocele, meningomyelocele, etc. Okay, so here you can see sometimes a baby may be born with an appearance like this, just some tuft of hair on the outside and no other visible abnormalities. If there is a lot of hair like this on the back in a bunch, then you suspect spina bifida occulta. Occulta means hidden. So the defect is hidden below, and usually there is only a vertebral defect, but the spinal cord and meninges are in place, and this may be accidentally discovered on an x-ray, or if you suspect it and do an x-ray if you see a tuft of hair like that. So there is no displacement, no neural deficit, and you may surgically repair it, or sometimes a person may not even be aware of this defect. The second type of slightly more severe defect is a meningocele, means there is a vertebral defect and part of the meninges have come out as a cystic swelling, so covered by skin. So there will be a swelling at the back and it will be soft to the touch, but there won't be any neural tissue in it. There might be only meninges. So then it's called a meningocele. But if neural tissue of the spinal cord also comes out through that defect and forms a swelling, then it's a more serious complication and it's known as myelomeningocele or meningomyelocele, where spinal cord also, a part of it has herniated. Obviously, this will lead to more severe signs and symptoms. So here are some examples where you see wider defects like that, means lot of multiple vertebral defects, and the whole neural plate is exposed either completely or partially. That is known as racheschisis. Racheschisis means split, like a split spine, okay, where the whole neural plate is exposed. So this is the summary of the third week of events. You can understand the most important events of gastrulation, that means formation of three germ layers, formation of notochord, and formation of neurulation. And this is all the summary that we've already done for you to read. Okay, how the primitive streak forms, first cells fuse with endoderm, replace it, then rest endoembryonic mesoderm, remaining cells ectoderm, then new notochord and neural plate, neural tube, etc. And then last is the sites where mesoderm is not there in the midline, at the site of cloacal membrane, oropharyngeal membrane, and where the notochord is. Okay. Lastly, the last thing to happen also is early vasculogenesis. This means what? Vasculogenesis means generation of blood and blood vessels. Okay. Now, how does that happen? So, notice in this picture, uh, that's the yolk sac and the amniotic cavity and the germ disc, etc., in the middle. Now, remember, I told you from the yolk sac, a little diverticulum, this is known as allantoic diverticulum or allantoa, has grown into the connecting stalk, which is the future umbilical cord. Now, this around the yolk sac and around the amniotic cavity and in the connecting stalk, remember, was extraembryonic mesoderm. So, in this extraembryonic mesoderm, this mesoderm, wherever there is mesoderm, intraembryonic or extraembryonic, gives rise to all supportive tissues. Especially in the embryo, it will give rise to blood vessels and other supporting tissues. In the next class, when we see derivatives of germ layers, you learn more about that. But the first blood vessels form in the connecting, not in the embryo proper, but in the connecting stalk around this allantoic diverticulum. See, you can see the first blood vessels, and slowly they spread in the extraembryonic mesoderm around the wall of the yolk sac as early as 3 weeks of development. Now, how does this happen? So, it's again in the mesoderm, groups of cells again get together. They are called blood islands, made up of cells called hemangioblasts. Okay, you can see these dark collection of cells, hemangioblasts, which are precursors for both. They are the early blood vessels, means they will form the wall of the blood vessel itself, and the blood cells themselves, which are inside the blood vessel. So hemangioblasts, which have derived from mesoderm, which first develop in the mesoderm around the connecting stalk. It is extraembryonic mesoderm around the connecting stalk. Later on, it spreads in the extraembryonic mesoderm around the yolk sac. Now, these hemangioblasts will differentiate into hemopoietic stem cells. What does this mean? These are the blood cells themselves, cells of the blood, early hemopoietic stem cells, and angioblasts, which get arranged around the periphery. So first you get mesoderm, just collection of cells. Then these cells are getting organized. Some remain at the periphery to form the angioblasts, and the others together the cells are known as hemangioblasts once they differentiate from mesoderm. Now some hemangioblasts become angioblasts, which surround they become a tube-like hollow structure, and other cells in the center will form the blood cells. So tube formation as well as their blood lining, I mean the endothelial lining and the wall of the blood vessel formation happens from the same group of cells or hemangioblasts. Now how does this happen? So under the influence of all these factors, vascular endothelial growth factor, etc., fibroblast growth factor, receptors, and FG2, FG1, don't worry about them. But obviously, it's under very precise control, right? Now, so here we see how blood vessels form again. First, as I told you, wherever there's mesoderm, but first it starts in the mesoderm around the connecting stalk. Some cells get together, they are known as hemangioblasts. Some hemangioblasts differentiate and form the outer layer of cells, which are going to form the wall of the blood vessel itself. And other cells in the center form the early hemopoietic stem cells themselves, or the blood cells themselves, right? Now, blood vessels arise in two ways. One is by the process of vasculogenesis, means the blood vessels, this what I've just told you in these upper pictures, blood vessels arise from the blood islands or groups of blood cells or hemangioblasts. The second next process to happen is known as angiogenesis, where further blood vessels arise by from existing blood vessels by branching or sprouting, growing out, as that is shown in the lower picture. So you can see early main blood vessel would have formed by early vasculogenesis, then it is growing branches, so that is formed by further angiogenesis. Okay, but you can see again, these are, if again, if excessive growth happens and it doesn't regress, you can see babies, sometimes you have seen babies with red birthmarks like that, mainly in the face region, or sometimes even growth like that. And look at this big growth in the third picture, something growing out from his mouth. So these are abnormal processes of vasculogenesis or angiogenesis, can result in tumor like masses known as capillary hemangiomas. Again, this is early embryonic tissue, so they can proliferate. Of course, they have to be operated soon, and everything can be corrected. Depends on the site. And these are common in craniofacial regions. Sometimes they may remain as birthmarks like that. Then they are not touched because, I mean, it doesn't do anything. But if it's like a mass obstructing something, then it has to be removed. Okay. These will be obviously very vascular because they are capillary hemangiomas because of the abnormal growth of blood vessels. Both the processes going wrong. All right. So that brings us to the end of this part. So where were we? We were in the remember, 1 to 2 weeks, early terminology is known as a pre-embryo. So the blastocyst, while it's implanting, is all pre-embryo. Then when the germ disc is formed, bilaminar to trilaminar germ disc, in the third week we've seen some changes. So that's the beginning of the embryonic period. We shall see next time what happens in the embryonic period, how the germ disc becomes a proper, like a folded embryo, and a a three-dimensional structure like that, and then go on to the fetal period. So we will continue the story next time. Thank you.